This video is based around the practical to investigate the growth of leaf yeast. It contains only the key revision points, things that we've encountered frequently, so it's serving as a revision prompt just before the exams. Yeast is a unicellular fungus, and there are many species of yeast found commonly on the leaves of plants in Ireland. Leaf yeast grow on the top and the undersurface of leaves, and the leaves that you take for this practical should be taken from the base of the shoots later in the year so that they've had time for the colonies to grow. Leaves should be collected on the day of the practical in fine weather and placed in a box. Leaf yeast is sensitive to pollutants in the air, so those leaves collected from highly polluted areas will have fewer colonies of leaf yeast on them. So the aim of the practical is to grow leaf yeast from leaf samples collected from different areas and to compare them, and the ash tree is an ideal plant. The colonies of yeast will grow on malt agar. Agar is a growth medium, but it had the nutrient malt added to it, very important. The control for this practical was a sterile petri dish with malt agar, but it was unopened. We did not add any leaves, and this was to prove that any yeast grown are from the leaves only. So your control plate is simply just sealed with parafilm, and now it's on to the one where you're going to place the leaves or the sections of leaves. So you take the sterile petri dish and it contains that growth medium malt agar. So next, using petroleum jelly, either whole leaves or leaf discs are attached to the lid of the petri dish with the undersurface facing downwards because the spores will be projected onto the agar. All of this is done using aseptic technique, doing everything possible not to introduce other microorganisms. When you're dealing with agar plates, sterile agar plates, you have to apply aseptic technique, and the first stage of this is always washing your hands. Anything to do with microorganisms, you always have to wash your hands. But another practice we always had to do was to clean all the surfaces with disinfectant prior to the experiment, flame all utensils that ever went near the dish, and we never fully opened the sterile malt agar dishes if we can help it. There was another additional precaution that we had to know about. The dishes were placed in an incubator at 20 degrees Celsius for a number of days, but after 24 hours we had to invert them, turn them upside down to protect the yeast from condensation. So what are the results? Well, in the dishes that had the leaves or the leaf discs, you should see distinct pink colonies of yeast, and it's important to describe them as being pink. And if there are many colonies, it generally indicates better air quality. How do you dispose of the plates? That's very important. Well, we sterilise them with disinfectant, and remember, sterile means nothing living. So there is another YouTube video which goes through all of the important steps. Please make sure you watch that. The very best of luck.